Hi, this is Miss Litton, and this is my wonderful, excellent, really last moment put together 7th period support Friday afternoon. We're all exhausted, tired, can't wait for the weekend. 7th period support students, say hi. Hi. We are motivated, okay? And we want to review Chapter 6, Metabolism. Your metabolism is all your what? I'm just curious.
All the chemical activity.
Enzyme-mediated reactions of your body, exactly. And we're going to focus on energy and enzymes. Things should be coming to your mind already. Do a splashdown of what you think are the key things to know about energy and enzymes. Just start splashing some down. What are pull from your brain? Pull from your brain.
Yeah.
When you think of one, come up here and write it. I have 4 pens. Come on, everybody, think of one. Write it on the board. Hurry up. Grab a pen. Write one, and then go sit down and let somebody else write one. Don't write so huge that you're in kindergarten. This is free-form 7th period review. I'll talk about whatever's on the board. If you didn't write it on the board, I'm not talking about it. I'm kidding. I'm just kidding.
Oh, nice. My beautiful hand. It's energy.
My girl that pulled that surface area.
I don't know if I'm right or not, but
Oh, I spelled it wrong.
I was gonna do that. Can I go next?
You can write it the same time.
Oh, yeah.
You can write it the exact same time.
Catalase is enzyme, so that works. It works.
Exergonic.
Catalase is enzyme.
Oh, Endergonic. I'm not right. Hey, first time writing left hand. Wait, hold up. Like, I'm just gonna write proteins because that's amazing.
I like it.
Just enzymes in general.
Okay, wonderful. Okay, so the first thing I want to talk about that I don't see up here is energy. What kind of energy is there? Tell me, what's this energy?
Kinetic.
Kinetic. What am I doing? Moving. Energy has to do with motion. And then if I had, let's say this was a snack of potato salad. It's not. What would this be? Potential energy. A form of, what's another word for it? Chemical energy. And we know energy is stored in bonds. We talked about that today too, right? So those are our different types of energy. The other thing I don't see up here are these, is this right here. You know what I'm going to write?
Laws of Thermodynamics.
Dynamics. Good. That's the other thing I don't see up there. What is the first one? What is the second law? The first law of thermodynamics says, wait, hey, do it together. What's the first law? Energy cannot be created or destroyed. It can just be transformed. The other name for it is?
The law of conservation of energy.
Boom. Okay, so that's the first law. Can't be created or destroyed. It can just be transformed. The second law of thermodynamics says what? Every time it changes form, yeah, it's not 100% efficient. Absolutely. So we covered energy, we covered the laws of thermodynamics. I feel good about that. Do I see the word entropy up there?
No.
No. Entropy would be another word, and that measures the level of disorganization. So when we look at this, we say this has a high level of entropy, a high level of disorganization, but it's very what? Stable. And the reason why it's stable is it really can't what? Yeah, it can't change, but you can't do much with it. Whereas the opposite, low entropy, right, has a lot of order, but it's not as stable. Good. All right, and then we applied that to things like solid and liquid water, right? Okay, and we talked about a solid as it breaks down, right? The molecules as it breaks down and creates farther and farther space between them, then you're going to have increasing entropy. And things always go from a higher entropy to a lower. Then how do you explain all the changes on this Earth? Yeah, overall the universe is going to greater and greater entropy, but the sun's entropy, its breakdown releases wavelengths of energy, which we can harvest. Who does the harvesting of that energy for us? Plants doing what process? What are their solar panels called? Be more specific. Chlorophyll, the pigments, right, are able to capture that light energy and turn it into what? Chemical energy. Good. All right. So as a planet, we are going and harvesting that energy in order to order our systems. But as we use that energy, as we use what the plants have made, we're giving off what? Heat energy in the process. So then we talked about the difference between glucose, right, and carbon dioxide. Now we know if we make this reaction go from left to right, if glucose is our reactant and CO2 and water are our products, what reaction is that? What was that? Cellular respiration. This energy that is given off when you break these bonds, we're going to use it to make what? ATP. Do we harvest all the energy out of the glucose molecule?
No.
No, we don't harvest all the energy out of that glucose. So we lose some as heat, which is part of what law? Second law of thermodynamics. Okay? And then we also talked about this, right? And we talked about it several times today. When you concentrate hydrogen ions to one side of the membrane, that would be low entropy, right? Because it's really high ordered, it's all on one side. And we know things want to go increase entropy all the time. So when we concentrate the hydrogen ions on this side, it's a difference threefold. What are the three things?
pH, concentration, charge.
Okay, concentration, pH, and charge. Is that what you said?
Yeah.
Okay. So it wants to go, and it wants to go to greater and greater disorder, increasing entropy to balance out. And in the process, when those hydrogen ions go from side A to side B, they could go through something called ATP synthase complex. So you can harvest that difference, that electrochemical gradient, and couple it with the formation of ATP. Yes? Okay. So now we know why we want to create this hydrogen ion gradient. Yes?
So when someone dies, would that mean that their cells have all used up, all of their cells have equal amounts of, equal levels of stuff?
You mean the cause of the death or following the death? Like rigor mortis?
I have no clue.
Well, let me give an example. Let me give an example. Only because you concentrate calcium to one side of a membrane in what's called your sarcoplasmic reticulum in your muscle cells. And then when you release the calcium, it allows these actin heads to bind to troponin, which moves tropomyosin, which allows your muscles to contract, okay? But you can't do that. You have to move it. You have to keep contracting, 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 contracting. And when you die, all the calcium just dissipates and you can't move it anymore. And so whatever stage, like if they fell off a cliff in a weird, body, you can't move it because the muscle becomes like paralyzed in that spot. Because it's that differential, and then you release it, and then you allow the muscle to move. Yes?
Wait, so if someone dies
Did you
in a
just say
certain,
wait? Did you say wait? Go ahead.
if someone dies in a certain position, you can't move them?
It's very difficult to move them. I'm sure
Oh,
they
I
just
figured
make
you
some
just, like,
cuts.
take their arm.
No, you can't. They will. At first you can, but not later. But we'll let that go for now. Yes.
I mean, regarding just natural causes.
That's another whole discussion on
Oh, really?
disease. Yeah,
Okay.
there's a lot there. Yeah. Usually if it's something due to cancer, it's because the cancer has uncontrollable growth, and as a result, it's interfering with other organs' ability to do their job. So as a result of that, you're out of homeostasis. And that can cause death. Yeah. Now, lots of biology there, but we'll keep moving on. Let's jump into, we know reactants, products, right? And then let's get in. Thank you. Gives free energy. Delta G. When the big ticket item here to know about delta G is when you have a positive delta G, when your amount of energy goes up positively, you must have done what kind of reaction? Because you would have to put energy into the system to make it go up. What kind of reaction is that?
Endergonic.
Endergonic. if your change, your delta G, your amount of free energy goes negative, you must have released energy, and that is exergonic. When you go to greater and greater entropy, greater and greater disorder, it's a spontaneous reaction, and you're probably going negative on your delta G. When you go to greater and greater order, decreasing entropy, then your delta G is positive. It's probably an endergonic reaction. Yes, those are the key things you need to know about exergonic and endergonic reactions and delta G. Okay, now the next thing we talked about was ATP being the energy currency, and that we know in order to synthesize, if we have ATP and if we actually break off one of those phosphates, if we spend that dollar, if we spend ATP, it's called hydrolysis because we're breaking that bond. Hydrolysis, right? Input of water. We snap off one of those phosphates, we will have the release of what? Energy. How much energy? Do you remember?
7.3
Boom. 7.3 kilocalories, right? I heard it from a couple of you. Good job. That's how much energy we'll get out. And so different reactions require different amounts of money that you need to pay to have those reactions go forward. But ATP is always worth 7.3 kilocalories. The reason why we do cellular respiration is to print more money, to hook the phosphates back on ADP to make what? ATP. Good. Question?
So I forgot what the exact percentage was, but how much can you use efficiently?
Remember how much usually gets passed on each time when energy changes hands? Do you remember that? Through producers to heterotrophs. Do you remember?
10%.
You haven't learned that, 10%. But glucose, you already gave me the number correctly today.
Okay.
Yeah, you did. So when you do cellular respiration, how much energy do we harvest out of the glucose? 39%. That's with what? Oxygen. Without oxygen, we only get about 2 ATP out of it. So that is not as efficient. That's why we have to do it with oxygen. Okay, and we'll learn both those reactions so you see why that is. Then we talked about coupling endergonic and exergonic reactions together, a coupled reaction. A reaction that requires energy with an reaction that produces energy in order to facilitate it. So when we look at endergonic reactions, active transport, anything across the cell membrane, like the sodium, potassium pump, like this, what's this? Endo and exocytosis, right? All of those things are transport across the membrane. That costs you energy. Any cell movements, like actin and myosin sliding past each other in a muscle, like cilia and flagella moving something around, that's going to cost you ATP. And anabolism. What is anabolism about?
It's creating stuff.
Yeah, building. Yeah, building molecules. Synthesizing, like taking your glucose and synthesizing glycogen. That would be an anabolic reaction. Hooking amino acids together to build a protein. That would be an anabolic reaction. So you need energy to do that, so you'll spend your ATP to do those endergonic reactions. You'll couple it with it. Conversely, when you do cellular respiration or break other molecules down that release energy, you can harvest that release in order to build more ATP back up. Questions, concerns, issues? Surviving, breathing, take a breath in. Do some cellular respiration. One more, feed your brain, feed your brain. Okay, now try this. Try, Jojo? Run out and see if Mr. Martin needs me right now. I can take a little quick break here. Just go, they're right, they just opened the door, they were looking for me. Oh, it's not Mr. Martin, Mr. Buchanan. Okay, all right. When we talked about metabolism, okay, when we talk about metabolic pathways, they are very, very, very complex, right? And we're going to learn about some of these. Don't think about memorizing. There's nothing here for you to memorize. But if you just like it, take a picture of it. It's super cool and look for it. Exactly. When we talk about metabolic pathways, the key thing is, do you see how many little steps there are? And that's because it's more efficient. You release the energy out slowly of each one of those steps. Also, it allows for more control because each one of those steps is going to be controlled by a what? What's the name of this chapter?
Enzyme.
Thank you. Okay, it's going to be controlled by an enzyme, and that's a control point, right? And so we talked about the role of enzymes. And enzymes are made primarily out of what? Proteins. But there are RNA enzymes called ribozymes that we'll learn about as well. And enzymes will facilitate a reaction, whether it's an anabolic or catabolic reaction. It'll facilitate it by bringing the reactants close enough together so that you can get your product. Now, the specific part of the enzyme that is key is called the what? Active site. And the active site is specific to the substrate. If you change the active site in any way, you will change that enzyme's functionality. Because if it can't bind to the substrate, you can't help it can't help facilitate any reactions. So controlling the active site is key. Now, when the enzyme is bound to the substrate, it's called induced fit. Induced fit. It may change its shape just a little bit as it binds to get that perfect kind of binding. Okay? That perfect inner binding at site or arrangement, I guess. So that's a slight, when it's binding, you call it enzyme substrate complex, or induced fit is how it binds specifically to that substrate. Its change in shape is very temporary. It's like it has the reactants, like if I was trying to make two phones come together, right? It has the active site, which is my hand, but to facilitate, I might change my shape momentarily. I have now made the two-phone complex, and then I release, and I'm able to combine other phones and other phones and other phones. Okay, so it's just a temporary change, I only need a little bit of enzyme because it can be reused again and again and again. You saw that in your reaction, right? What was your reaction that you did? You did a reaction in your lab.
Catalase H2O2.
H2O2 and we put catalase in.
What did you generate?
Water and oxygen.
Right, that's not balanced. Okay, now, how did you measure that reaction? Did you measure the amount of substrate that was used up or product that was produced? Which product did you measure? You measured the amount of oxygen. What did you use to measure that amount of oxygen? Pascal probeware, right? And you measured it in parts per million. Why am I asking you these questions? I'm asking you these questions because when you review for your quiz, I also want you to be writing your
Lab.
Okay, yes, lab. But what else do I want you to prepare for? Essays. And there's an essay question on enzyme structure and function and a lab to support that. Okay, so look over that essay because this is the lab you would use. You don't have to, on the AP exam, create some new lab you've never done before in order to explain something, just to show how smart I am, okay? No, use the labs we did, okay? Verbatim, okay? Read their question and answer it. They may bring up this same reaction right here, but they may say, how could pH alter this reaction and how would you measure it? You could still do the same reaction, but where we altered the substrate concentration, the amount of hydrogen peroxide, what could you do?
Alter the pH.
Alter the pH that the catalase was in, right? You could add catalase with no sulfuric acid, catalase with 10% sulfuric acid, catalase with 20%. Do you see what I'm saying? And so you could see how the pH affected the catalase. If they asked you to do this with temperature, you would alter the temperature that the catalase was put in, right? At, 0 degrees Celsius, at 20 degrees Celsius, at 40 degrees Celsius, at 60 degrees Celsius, and see how that affected the reaction. How would you know how it affected it? By measuring the amount of oxygen produced, right? There's lots of ways we could change this reaction. Yes?
At one point, what is the temperature denature the protein though?
Exactly, and so you could find out when that is, right? You could keep upping it by 10 degrees until you found where you got no product at all. And you would know, well, it denatured. Because you would expect the amount of product you have or the rate of production to change, right? Okay, so I just want you to keep in mind, read over your essays, use that lab that you did in order to write that essay. In your pre-lab, I asked you to talk about the structure of an enzyme and how it impacts its functionality in the active site. That's just what I asked you for also in your essay question. So write it, get it done. Write it for this chapter, because this is the first chapter of your new unit, right? Wouldn't it be great to write the essays? Could you go find an answer to this essay? Google it, you'd have one in a heartbeat. But then that's no work for you. That's like running a race and getting in a car instead of running the race and showing up at the end in your car. You have no ownership, you have no training, and when you're asked to run that race, you don't have the conditioning. You got to run your own race. You can train with somebody, run with them to help motivate you, but you're each going to have to be able to run your own race, so figure out how to write it. Yes?
Where did you say we're finding those essay questions?
Where do we find those essay questions, Unit 2? On my website. Where?
Under
Resources for review.
Resources for review, and it says potential essay questions.
And it goes by chapter?
I list all of them that it could be, and then I have them in sequence.
Cool.
Okay, now we know enzymes are named for their substrate, right? And we know what enzymes basically do is they lower, what did we call this right here?
activation energy.
Yes, energy of activation. It lowers the hurdle, so it makes it more likely that the kinetic energy of the system itself is enough to facilitate that reaction. Okay, it lowers that. And we looked at it, you know, with an enzyme, without an enzyme. You know, what is it, will it happen? If we let hydrogen peroxide sit out overnight, some of it will break down into water and oxygen. It will just spontaneously break down. But how many more times faster can it be with an enzyme?
10 million times.
10 million times faster with an enzyme, exactly. Okay? We talked about anabolic and catabolic reactions, and we're good on that. Now, factors that affect enzyme activity. We've already talked about one, okay? And you did a lab on the very first one. You did substrate concentration. And I'm betting in all of your labs that 3%, that your reaction rate was much higher at 3% than it was at 1.5%. Is that accurate?
Yeah.
Okay. But we also talked about pH. And the every enzyme has an optimal pH that it works best at. Which one of these is probably found in the stomach?
Pepsin.
Pepsin. And that's where Pepto-Bismol comes from, or Pepsid AC, because this is the enzyme that is in the stomach that breaks down proteins. And so it works at a low pH. When your food moves from your stomach into your small intestines, the pH is neutralized there because the small intestines are not prepared for that low pH, so it gets immediately neutralized. Pepsin won't work anymore. Instead, you need trypsin, and chymotrypsin will help, digest your proteins then. And we talked about enzymes having an optimal temperature where they work best. And we talked about organisms that cannot maintain their body temperature, then their enzymes are not as functional, and that's why their activity is based on temperature. Whereas we have organisms that are endothermic, then they're functional at all times. All right, unless they're tired and hungry and grumpy. Siamese cats, we talked about cofactors, an inorganic cofactor. It could be like an iron or a metal that activates it. If it's organic, it's called a coenzyme. You're not going to say it's a protein, because if it's protein, it's just part of the enzyme, right? Because enzymes are proteins for the most part. And then we talked about, there's a coenzyme, great vitamins. Two types of inhibition shown here, competitive and non-competitive. Competitive, it's competing for
Active site.
the active site. Something else wants to bind onto the active site. It could be reversible, it could be irreversible. If it's irreversible, it's probably a poison, okay? But it could be a way to control when it binds on. And non-competitive inhibitor binds to a secondary site, referred to as the allosteric site. Is it on here? Is it listed allosteric site? No. But this secondary site right here, when it binds, when the non-competitive inhibitor here, it still affects the active site because it changes the active site, the shape of it. Because you know how bonds will rearrange themselves based on something that's happening here. They'll keep rearranging themselves, which can end, in turn, it could affect the active site. It kind of like if you had a giant beanbag, right? And it was situated like so somebody could sit in it. But if you go to the other side of the beanbag and you push on it, right, it's going to change the seat, right? This is going to influence that. You didn't actually touch the seat to change it, but you touched some other part that influenced it. Yes?
When you say poison, does that mean that they denature the active site, or is it just...
It just means that it binds on, like I told you, carbon monoxide will bind on, and then oxygen can never get on there. So it basically shuts down that whole hemoglobin system there. All right. Then we talked about, and this is not a bad thing in any way, and a lot of the inhibitors are not bad. I'm just giving you the extremes when I talk about a poison. These are ways to control reactions in those metabolic pathways. And in end product inhibition, what happens is you could have this metabolic pathway, A to B to C to D to E, etc. This long metabolic pathway. You don't want to keep synthesizing all of these things and then at the end you go, oh shoot, we don't need this anymore, because then you have all this extra buildup, right? So stop it before it starts, right at the beginning, that very first reaction. If you have enough of product, whatever this product is, if you have enough of it, it binds to the very first enzyme, so it disables that enzyme so you don't start this reaction until you run out of that product. So here we can see, here's the first reactant binding to enzyme 1 right here, okay? And then it's going to make, sorry, I'm giving myself a headache, A to B to C to D to E, and here's your end product. If I have enough of this end product, it will bind to enzyme 1 and change its active site. Remember the active site before? Smooth. Reactant A can bind onto it. Here, it's changed it. So then you stop this whole pathway by just putting a stop here at the beginning. Okay, and I don't think I did these questions. I didn't have time for them in class, so I wanted to do these right now. So I would be, your favorite thing you're looking forward to this Friday afternoon is how I will have you log in. You're looking forward to an electron transfer chain and amniotic fluid. What is yoinking? That makes me uncomfortable.
Yoinking is stealing.
What is it?
Someone put dying.
Why am I?
Oh!
What's going on?
For some reason it like automatically.
Wait.
An iced caramel macchiato. Good choice.
I think put I'm pretty sure yoinking was in your
I thought you were big sound.
Okay. When you grab
It's like the grabbing sound.
Okay, go. Do these questions. I'm going to pause this. Remind me to restart it. 3. Are you ready? Commit. We have some zeros in here. 5, 4, 3. Eating chicken. Tacos. I love tacos. You better not be drinking font. All right, let's talk about it. You guys, cállate, mon ami. Focus. Here. Okay. The active site of a specific enzyme is similar to that of any enzyme? No. They're specific to their substrate, so definitely not A. Is part of the enzyme where the substrate can fit? Yes. Can be used over and over again? Yes. Is not affected? They are affected, right? Some of you just picked B, but the rest of you got everything else. Good. Oh, if you want to increase the amount of product per unit time of an enzymatic reaction, did you read what this said? Do not increase this. Did you see that? Do not increase this.
So the pH does not.
Okay? If you increase the amount of substrate, it says do not do this. If you want to increase the amount of product, don't do this. If you increase the amount of substrate, will you increase the amount of product? Yes. If you increase the amount of enzyme, will you increase the amount of product? Yes. If you increase the amount of temperature somewhat, will you increase the amount of product? Yes. If you increase the pH, will you increase the amount of product? No. Every time you change the pH, it's a tenfold change, right? Tenfold concentration. You change pH, you're going to decrease the amount of product that you get. Yes?
Sorry, how would temperature cause a difference?
If you increase temperature, notice the key word here is somewhat. Increasing temperature increases kinetic energy, increases the likelihood that that reaction will take place.
Thank you.
All right, number 3. Whoops. The diagram depicts an, oh, dang it. There we go. A non-competitive inhibitor. That answer is what? True. Why is it true? It didn't bind to the active site. Because if it bound to the active site, it would be a competitive inhibitor. It bound to a secondary site. And then 4, an allosteric site on an enzyme is the same as the active site? No. It's the second one. Is non-protein in nature? No. It's on the enzyme, so it's protein. Where ATP attaches and gives its energy? No. Often involved in feedback? Yes. Because that, remember I talked about where the product, if you have enough product, it goes back and binds to that allosteric site and changes it on enzyme 1 so you don't have that reaction go forward. All right? Now, this brings us to what we talked about today, so I should be able to go through this pretty quick, right? Reduced and oxidized. If I was going to summarize ways to reduce something, what could I say?
Gain energy.
Gain energy. What else could you say? Gain electrons. What else could you gain? Hydrogen or lose oxygen. And it's the opposite, right, for oxidation. This is a way, when we look right here, it's a way, whoa. It's a way to transfer energy from one molecule to another. By me giving you my electrons, I'm losing the energy and you are gaining the energy. Did I remember to hit play? I did. Okay. So then we took that to photosynthesis and cellular respiration, right? And we brought it down into here. And we said here, when photosynthesis, when CO2 becomes C6H12O6, what has happened to the CO2? It got what? Reduced. Okay? Whereas water gets oxidized. Where do the hydrogens come from to reduce the CO2 into glucose? Right there. Right? The hydrogens from water will be used ultimately to reduce the CO2 into glucose. Who's going to carry those electrons temporarily? Where are these hydrogens going to go temporarily? They're going to go to who? NADP. Yeah. They're going to reduce NADP. Who's going to give it to CO2 in order to reduce it, right? Now, if you're going to be doing that, right, in order to form NADP, you need some high energy electrons to form NADP. Who's going to get those electrons excited? The sun. So that's why the sun is a reactant in this whole process. Yes?
Yes.
Okay. And then we talked about in photosynthesis, we need oxygen, right, to do what? What does this oxygen do when it gets reduced? It has to take the electrons at the bottom of the electron transport chain, right? And when it does that and hooks up with some hydrogens, it forms water. So it got what when it becomes that? Reduced, right? And where was the source of those electrons to put through our electron transport chain? Right here was the source of those electrons, right? So we rip the electrons away from glucose, and then we're left with just CO2 as a metabolic product, as well as what do we make? ATP. Good. So we did that, and that should be pretty fresh in your head. And in order to achieve that, we do an electron transport chain, which is a series of redox reactions which enable us to set up, ready for this? You can do it. A chemiosmotic gradient.
Chemiosmotic gradient.
You just repeated me. You're so cute. You're like parrots. Okay, and yes. Same thing. It's an it is one and the same. You're fine. So this chemiosmotic gradient is allowing us to have a difference in what three things? pH, concentration, charge. So we can couple that with the synthesis of ATP. Got it?
Got it.
All right. Good job, guys. I'm very proud of you. Easy peasy lemon squeezy. Don't eat food that is too greasy. If you're hungry, have a piece of toast. I love you the most. All right. Don't stay up too late.